Hi everyone, welcome to this talk. This talk is about using scientific Python to analyze X-ray images of metallic alloys undergoing solidification. We are two speakers, so we want to take a minute to introduce ourselves, and then we'll dive into actual image analysis and material science.
I'm Gus Becker. I'm a PhD candidate in material science at Colorado School of Mines. I'm a part of the research group CANFSA, which is the Center for Advanced Non-Ferrous Structural Alloys.
I'm Marianne Corvellec. I work as a scientific software developer, and about 6 months ago, I joined the core team of scikit-image. I will be presenting scikit-image in just a little while. And I'm also an independent researcher. In this respect, I am affiliated with IGDORE, which stands for the Institute for Globally Distributed Open Research and Education.
So before we present on scikit-image, I'd like to talk a little bit about the material science context for the experiments that we'll be analyzing. So what we see here is a diagram of a laser additive manufacturing process in which a laser melts metallic powder to create a part or an object. And through this process, we're able to create parts with more complex geometries than are possible through more conventional manufacturing processes. However, through the laser metal interactions, we achieve much higher thermal gradients and much larger solidification velocities than you would typically get in, again, in these conventional manufacturing processes. Since these processes are relatively new, studying the solidification process as it occurs is of interest to understand the phenomena at play and what kind of final microstructures result from these additive manufactured parts.
So scikit-image is distributed as a Python package. Therefore, you can install it with pip or conda, and I recommend that you work in a virtual environment. Please refer to our website, scikit-image.org, for instructions on how to install the software package. If you're interested in the website, you will also find instructions on how to contribute to the project. Once you have installed scikit-image, you can import it. First, you will have to launch your favorite Python IDE or code editor. I recommend that you start a Jupyter Notebook because it's particularly suitable for exploratory data analysis, and more generally, it's a very user-friendly and versatile tool. For example, this very slideshow is based on a Jupyter Notebook. So, how can we get started quickly? Scikit-image comes with a data registry. By importing the data submodule of scikit-image, you have access to many images that you can readily use to try out the various image processing algorithms implemented in the library. We have sample photographs, a cat for example. We can see that an image for us is simply a NumPy array. This means that we can leverage all the powerful machinery offered by the scientific Python stack. Here we can see that the NumPy array has 3 dimensions. That's because we're dealing with a 2D RGB image. So we can see that we have 300 pixels in the row dimension, 451 pixels in the column dimension, and 3 color channels. So let's take a look at this cat picture. I'm using the imshow function from Plotly to display my image, so the display I'm getting is interactive, as you can see. For example, if I hover on a given pixel, I can read its coordinates as well as the intensity values for each color channel. A typical problem in image analysis is that of segmentation. That is, when you want to separate structures from a background or from other structures. In this image, we could be interested in segmenting this light structure as well as this darker structure in the center. If we consider channel number 2, which would be the blue channel, we can see that multi-Otsu thresholding works well for finding the darkest structure at the center. Multi-Otsu thresholding divides up the pixels of a single channel image into 3 classes by default, hence separated by 2 thresholds, based on the distribution of intensity values. If we apply the same kind of filtering on the other 2 channels, R and G, I'll let you try it at home. You'll see that we don't pick the other structure of interest, the light one. Yet, if we simply change color spaces, going from RGB to CIE lab in this example, we can see that the same thresholding applied on the first channel does yield the desired segmentation. Of course, it would take other and further filtering steps to smooth the boundaries of the thresholded regions. Then you would typically label the different regions and measure some properties for them, such as their size or the position of their centroid. You will see that these utilities for labeling and measuring region properties come into play in the material science application, which Gus is going to present right now.
Okay, so now I'm going to load an additive manufacturing simulator data set that was captured at the Advanced Photon Source at Argonne National Lab. The Advanced Photon Source is a synchrotron user facility that allows us to capture in situ solidification data using transmission X-radiography. So I'm going to load this data set using the IO submodule of scikit-image. We're going to load an image collection of the image files in this directory. We can see that there are 11 of them. When we do the shape, we can see how many rows and columns are in each image. In order to get a stack of smaller images, we're going to crop each image in the data set. Then we can use the animation functionality of Plotly to show an animation of the sample solidifying directly after the laser shuts off. So when this runs, you can see the solid-liquid interface here solidifying inwards and shrinking as the region that's liquid starts solidifying. So the first thing that we'll do is import some custom local modules that contain some helper functions, both for plotting and for processing our images. So first thing we'll do is plot the raw images by applying a color map of purple to yellow. You can see the solid-liquid interface getting progressively smaller throughout these images. And then the first processing step we'll do is apply a Gaussian filter to these images. You can see we're loading up the first image here, and then we're going to subtract the current image from the succeeding image that we just loaded, and then plot each of those images. And then what we're seeing here we'll call the image delta, the difference between one image and the succeeding image. And what that shows us is the distance that the solid-liquid interface has moved from one image to the next. The next thing that we're going to do is clip the highest and lowest intensities of these images, and that is going to brighten the light regions and then darken the dark regions. You can see we're adding track.clip here. What that's doing is taking the top 5% of intensity values and just shrinking them to a single value, and then same for the lower 5%, just shrinking them to that value at that 5% max intensity. Then we'll simply invert the image, which turns the bright values to the region of interest, which is the solid-liquid interface, and then makes everything else that dark region. And then we'll, using the restoration submodule of scikit-image, we'll denoise the images. So you can see here restoration, and then we're using this denoising algorithm, applying a certain weight and then returning that image. When we plot this, you can see compared to the previous image processing step where we have quite a lot of noise outside of the solid-liquid interface region, we can now see that it's generally a lot more smooth. This sets us up to threshold the image. So we're going to perform a simple thresholding operation at some thresholding value, in this case 0.4, and then return the mask or the binary image that we create from that thresholding. And you can see here now we only have an image mask containing ones and zeros, ones for the yellow and zeros for the purple. So the next thing that we want to do is look at our binary image here and try to select this larger region from all of these smaller regions. And the way that we'll do that is using the submodule from scikit-image measure. First, we'll call measure.label to create a label of our image mask. And then we'll call color.label to RPG to convert our labeled image or at least convert the labels of our image two colored labels to overlay on top of the image, and that's on top of the raw image. And when we do that, we can see the different regions. The next thing that we want to do is look through each of these regions and assess some of their properties. So using measure.regionProps table, we can look at specific properties. Right now we'll look at the label, the area, and the B box, which stands for bounding box. So label increments up, and then we can see the area. There's certain regions that have larger areas than the other regions. And then B box has our minimum column, minimum row, minimum column, maximum row, maximum column. So to see this more clearly, we're going to sort these values, sort the area in a descending fashion, and then only show the top 5 values or top 5 rows of our data frame. And when we do this, we can see that there's one area that's much larger than the other areas. So what we'll do is now using measure.regionProps, which creates a dictionary of these properties. We'll create a list comprehension to build a list of region areas for any region in this region dictionary if the area is larger than 100. And what that does is returns a list with just that single value of 229 for the largest area. And when we add this line to our image processing function with the minimum region size argument, we can see the single region in our images. So the next thing that we want to do is do the same thing, but instead of looking at the area, we want to grab the B box values. So when we do that, this time building a list comprehension with the region bounding box values, we get a 4 tuple inside of the list instead, corresponding to each of those B box values. And what we can do now is check that that makes sense. So minimum row is 0, maximum row goes up to 23, that matches, and then columns we have 17 to 61. So that makes sense. And now we want to add this to our processing function. Now with the keyword argument return bbox, and now we're capturing the bbox and returning it, so we can pass it to a plotting function and get this rectangle out of it. And when we do that, we can now send the same function to the whole data set. And we can see that we get a bounding box for each of these images. And it pretty closely matches the solid liquid interface. In some cases, it under-identifies, and in some places, it over-identifies the solid liquid interface. But especially as far as the lower bounds go, it's pretty good at identifying where the solid liquid interface exists. The next thing that we'll do is compare this automated detection to manual measurements, and these manual measurements were made using Nipari, which is an n-dimensional image viewer made with Python, and that can be used very fluidly with Jupyter notebooks. You can open up a Nipari window to look at a stack of images and then transfer. You can click on points and annotate an image and then pull those back into a Jupyter notebook for things like plotting or saving. So we can see here that although the detection is generally larger than the manual measurements, the trend matches, which is a good thing to see. So we do still need some further analysis and maybe honing in on some of these different ways where we can improve this, but generally we are matching the trend, which is a good thing.
Before we wrap up, we would like to thank the following people, organizations, and communities for all their help and support. Personally, my work with scikit-image is funded by a grant from CZI's Essential Open Source Software for Science program. We would like to point you to our public repo as well, where you'll be able to find all the materials that we presented in this talk.